new collaborations. So thank you so much.
Thank you, David. I'll just check to make sure that you can see the right screen and everything looks okay.
Yes, so we've got a full screen mode here, and it looks great, and we can hear you loud and clear. So this is phenomenal.
Perfect. Maybe I'll start with an important acknowledgement. I'm sitting here at UBC, which is located in the School of Biomedical Engineering, which is located on the historic ancestral and unceded territories of the Musqueam, Squamish, and Tsleil-Waututh peoples. And I'm really privileged to have the opportunity to share knowledge and information across the province today. So thank you very much for that opportunity. I'm gonna talk to you about our research, and it was really fun thinking about this talk, because I put together actually a new talk that kind of talks about how what the primary thing that my lab is interested in, which is blood development, links into how tissues are built, and how some of our fundamental work on tissue collective behavior might be of interest to this community. If we have time at the end, I'll make a few comments on the school of BME, but we'll see how we do with the research talk before we get there. So I did want to start by kind of linking these three images together, which I think are really wonderful to be able to present in Canada and in BC. And what it does is it shows the very early work, autoradiographic work by Clermont and Labonde at McGill University, where they identified the spermatogonia and how cell proliferation within this organ really led to this particular tissue formation. And they hypothesized that there was a special cell, a stem cell, which was capable of giving rise to this. This then links a number of years later to some of the really foundational work on defining what the properties of stem cells are, their ability to self-renew and differentiate. And I've picked a picture here from Jim Till, Ernest McCulloch, and Lou Siminovich, work that shows the colonies of blood progenitor cells of different types that formed in the spleen of these animals. And this was the beginning of trying to be able to quantify stem cells and what their properties were and how they behaved. And then, of course, in British Columbia, Connie Eves and many others have taken up this banner and really taught us significantly about the properties and how these systems are organized. And this is a very traditional organization of the hematopoietic or blood developmental system. And of course, we're learning more and more as we have both functional and molecular assays of the different cell types. So what I'd like to do today is try to connect a few different aspects that are going on in my lab. One is our efforts to understand how individual cells, primarily pluripotent stem cells, but other cell types as well, including T cell progenitors, make cell fate decisions by understanding the regulatory networks which are inside these cells and what the logic of that decision making is. I'd then like to connect this up to how individual cells that make decisions actually communicate with each other through collective behavior, and that determines the relationship between different types of cells within tissues, how they're organized and patterned. And I don't think I'll have much time to talk about today our more synthetic biology work, where we then take this information and try to program tissue fate and function, although there'll be some insights into this as I move along the way. So I'll start with some work that we published now, I guess 5 or 6 years ago, that was really based on the observation that we needed better ways to connect the environment around the cell to the decision-making circuitry within the cell. And this is a collaboration with Microsoft. This started with Sarah Jane Junn at Microsoft Research when she was there, and then also with the Systems Biology Institute, Hiroaki Katano and Ayako Yashi when she was there. And the basic premise is a cell receives a series of input signals. There's some processing information. This is both the signaling and the transcriptional processing, which leads to consolidation of a decision, which is expressed through a functional outcome. And how this input-output relationship actually occurred in individual cells has been very, very difficult to model because there's so many different ways that these things can be organized, and we're always limited by, can we actually measure this parameter and what this parameter means. So we've taken a different approach, a sort of rule-based machine learning type approach, where we identify rules that satisfy the constraints of the experiments which we're taking, i.e. if this is the input and that's the output, what are all the different ways that a network can be organized that satisfies this? And of course, as you add more experiments to this, you're able to reduce the constraint space till you start to have networks where individual cells can be predicted to act in certain ways, and then eventually how those cells would interact with their environment, as I'll show you in a second. So the latest iteration of this technology was just published actually last month, and it was some work led by T.M. Hidari, who is a PhD student in my lab, in collaboration with Matthew Langley, who's at Caltech, and Ayako, who's actually here, but still working for the Institute of Systems Biology in Tokyo. And they published a paper on a piece of software called IQCell. IQCell is able to take single-cell RNA sequencing data, automatically select informative genes along a time-based hierarchy. This can be either real-time or pseudo-time. It constructs dynamic gene regulatory networks from these, either using or not using a priori knowledge, and then will predict the effects of genetic perturbations on these differentiation trajectories. We developed this technology using the T-cell differentiation system. This is really for two reasons. One, because my lab is very interested in T-cell development, but actually another big reason is this is a well-studied differentiation hierarchy, so that we had good training data to help us understand what the constraints are in building this. And for those of you who don't look at T-cell development every day, of course, you have thymic progenitors, which undergo early differentiation. During this differentiation, in this case, I'm showing a mouse hierarchy here. You lose potency in different types of lymphoid cells along a developmental trajectory till eventually you end up with more mature T cells. And there is lots of single cell sequencing data, and here in this principal component plot, you can sort of connect the progenitor cells on one side. So I'll just make sure I have my pointer well available. You can connect the progenitor cells on one side through a differentiation hierarchy to the mature cells. We used IQCell to recreate this gene regulatory network, which is a simulatable or executable regulatory network, and many of the gene interactions predicted were captured in those based on experiments that have been done over many years. And our software actually predicts both T-cell development and erythropoiesis data with much higher fidelity than other software packages that are out there. We then used this to develop an in silico map of the dynamic simulation of the inferred gene regulatory network. Here is just another example of the experimental versus the simulation of those genes as they move through these pathways. And then this gave us enough confidence to take the next step, which is to start to do in silico knockout perturbations of developmental trajectories. And I'll just highlight this section of the plot here, where you might have a wild type network where gene A or factor A regulates B and regulates C, and you can overexpress one of those factors, or you can knock them out in this Boolean simulation of this aspect. In silico, we could then simulate where the developmental process either would go wrong or right, and make predictions about how these particular perturbations would take place. This allowed us actually to replicate most or all of the current knowledge on key genes involved in T cell development, what their effects were on those developmental hierarchies. And when we went back and tested both known and unknown components on this, we had very high predictabilities, with the numbers in red showing how we're able to predict the correct blockage in a functional way of different perturbations along that pathway, but doing this in silico directly just from single cell sequencing data. So for this section, I'll bring forward a few aspects that we're currently working on that may be of interest. We're now starting to link this information, which is really single cell information, to knowledge on how cells communicate each other. So instead of having the factors which perturb the pathways be, for example, cytokines or growth factors that are provided by an environment, we're linking this up to the factors which the cells are producing themselves. And then, of course, this is helping us to understand how cell-cell communication impacts these intracellular, intracellular, or between cell gene regulatory dynamics, to try to understand how these multi-cellular tissues might develop in this scenario. So with that background, that gives us a way to look at what the code is inside an individual cell as it develops a tissue. And we'll park that information for a second, and I'd like to talk about now how we can start to study tissue patterning itself. And I'll start with this image. So this image is an image of a colony of human pluripotent cells. What you're seeing is a colony that's patterned. And what I mean by patterned is we use microlithography to put extracellular matrix on the surface of a plate so that the cells can only adhere across this colony. You can see there's probably 10,000, 100,000, 50,000 cells here. And when we start the experiment, you'll see in a second, all the cells are equivalent. And then we add an inductive factor. In this case, we add the factor called BMP4, which is a cytokine that induces a feed-forward loop between BMP4 and other factors. And this causes Turing-like patterns, where you have then in the middle of the cell, you have SOX2 expressing cells that give rise to neuroectoderm. In the middle here, you have brachyury expressing cells that give rise to mesoderm. And then out here, you have cells that give rise to either ectoderm or extraembryonic endoderm. And this is a very reproducible way, and it's a very interesting phenomenon because you add a single factor and you get this beautiful reproducible tissue pattern from this. So I won't go into all the published work where we started to understand how this happens, but basically what you're looking at here is a summary of a series of papers, one published in Development and one published in PLOS Biology, where we unpacked the underlying regulatory mechanisms by which this tissue patterning would take place. And interestingly, it brings up a lot of the it brings together some of the early work, of course, by Jim Walbert and others around transcriptional signaling and how, for example, in this classic French flag model, different types of tissues might be exposed to different thresholds of signaling, which lead to different fates, with the bistable feed-forward loop that Alan Turing and others have proposed to occur to regulate how these different signaling moieties would be regulated across colonies and propagate across tissues. For example, in this system, we add BMP4. We know through the series of experiments that BMP4 positively regulates noggin, which noggin then negatively regulates BMP4, and BMP4 also has a feed-forward autoregulatory loop with itself. This creates these fluctuating regions of SMAD, pSMAD activation. That's a signaling pathway downstream of BMP4, which, if the colony is the right size, and here I show that colony being across this gradient, you get this fate activation, and then we can switch the fate by the addition of other factors. For example, if we add nodal, we get the mesendoderm pattern that I showed you here. But if we inhibit nodal, we get a neurectoderm pattern where we get a flipping of the types of differentiation that occurs here. Okay, so that gives us a nice bit of background on how we can start to pattern tissues in a very robust way from human pluripotent cells. But as you noticed, all those tissues are axisymmetric. But of course, tissue development often has bilateral symmetry or other types of symmetry. And so what we wanted to try to connect is how might this asymmetry start to emerge as tissues develop, using human pluripotent stem cells and these gastrulation-like models as an early system to understand this. So to more formalize that question, if we have a cluster of cells, either in 2D or in 3D, how do we start to get polarity and symmetry breaking within these systems? So this is where Daniel Aguilar Hidalgo, a research associate in the lab, comes in. Daniel is a biophysicist and was very interested in some of the underlying theory behind how signaling gradients form during morphogenesis of tissues. And in terms of background, what I showed you before is this, which is happening, which is you start with a homogeneous tissue layer of cells. You create boundary conditions that create a fixed input, what happens on the edge, and through this activator inhibitor loop which takes place, you get these gradients of signals which lead to gradients and patterns. This is really what we think has been happening up to now, but of course that doesn't give us the opportunity to have asymmetries emerging in this situation. So one way to do that is to change the boundary conditions. And this is an example where you might have fluid flow across a tissue, which creates a morphogen gradient, where you have a higher threshold of signaling on one side and a lower threshold of signaling on the other side. And people have done this using microfluidics and other things. And then you have high signaling here, low signaling here, and you've reorganized the sets of tissues. But of course, in the early embryo, we don't necessarily have fluid flow or sources of the asymmetries. So Daniel wanted to see if there was a way we could take away the need for this imposed boundary condition and have self-organizing boundary conditions within the system. And I won't go through the mathematics because they're quite complex, and time-consuming. But basically, what he did is he linked the boundary conditions also to the activator inhibitor feedback loops in a way that allowed us to explore how different sizes or concentrations of factors might create boundary conditions. And so what that allowed us to do was to define a phase space where I'll orient you. On one axis, you have the numbers of peaks of the different tissue types within a colony. And then on this, you have a normalized scale. This is the relative system size relative to the decay length of the inhibitor. And if you read across here, you'll see 4 different plots for each one. One is the 2D simulation of the gradient within that system, then is the data of signaling of SMAD activation, and then what that colony looks like in 2D. So here you have a homogeneous signal. All the cells receive the same signal. Here's a picture of those cells, and here's the mathematical model of that picture where all the cells are the same. They're all purple. This is what I showed you before, these donut-like ring colonies where you start to have gradients. You have these gradients forming differences in SMAD activation, which lead to differences of fate, which we can simulate. And then up here, you have these unusual things where your scale is so large that you have many different tissue spots emerging through these different peaks from this system. What was very exciting and unusual to us is that within this phase space, Daniel noticed that there were particular domains where the noise in the system matched the heterogeneity around the early signal initiation, and he was able to get reproducible asymmetries. Here you see that there's a tissue emerging here that has asymmetry with respect to where differentiation is occurring relative to these spots or these systems, or here you have a three-way asymmetry emerging here. So we then went on back to our experimental system where we could pattern pluripotent cells on plates, control both their density and their priming with growth factors, and show that indeed there were certain conditions where we had a very high probability that asymmetries in early tissue emergence would occur. And some of these asymmetries are shown here. With large colonies, you have more reproducible rings. With small colonies, you have more homogeneous cultures. And then in the middle, you have increasing probabilities of asymmetries. Just to quantify this here, we defined asymmetry as the difference in the ratio of the centroid of the signal which we were following versus the centroid of the overall colony. And you can see that at some colony sizes, a significant proportion of the tissues were asymmetric versus in larger ones, they were highly symmetric. And then we went on and really identified, and I won't go through all the data here, although I'm showing a little bit on the next slide, to really be able to identify all the different types of tissue patterns which could be formed as a function of length and activation scale within the system. This, of course, gives us a foundation for now not only understanding where asymmetry might emerge, but how growth might affect the changes in the activation and inhibition that occur along these tissues. So what Daniel is doing now, so far I've talked about, and this is actually a time-lapse movie of an asymmetric tissue. You see, in this case, we have I think it's brachyury labeled for in the early differentiation of the cells. They start homogeneous. The smaller colony becomes polarized with brachyury on one side and the other cell type on the other, whereas in the larger colonies, you have this donut-like ring. Now, if you have a growing colony, you have different ways that tissues move through these domains as they grow along, and we're starting to study how these activation thresholds might affect differentiation dynamics across multiple developmental states along this route. Okay, so far I gave you two pieces of information. One is how single cells interpret decisions based on the underlying regulatory network structure within those cells, and then how the environment and the feed-forward and activator inhibitor loops around tissues might pattern the outcomes. What we hadn't done, and what we were really excited to do, and this is unpublished work, although it's not for lack of trying, I must say, we're getting very close, is really, can we connect these two aspects and make predictions from the regulatory network structure of individual cells, what the 2 and 3D pattern would actually look like. And so this is work that was led by Himanshu Kaul when he was in the lab. He now has his own lab in the UK. And Himanshu used a version of our IQ cell to extract a executable gene regulatory network that helped us determine what the effects of signals around the cell, such as BMP, noggin, Wnt, and DKK, were on the underlying regulatory network structure of early pluripotent cell differentiation. This could all be done, of course, in a computer. And here I just show you a simulation of adding and subtracting cytokines to the environment in silico, and what the effects are of these cytokines on the genes that are expressed in the in silico cell. You remove BMP4, you get mostly SOX2. You add Wnt and you remove Wnt, you get non-OG BMP and SOX2, and so on, as these pinwheel plots show what the simulated effects of these cytokines are. Of course, as I said before, we wanted to link the gene regulatory network's structure to the colony which then emerged. And in order to do that, we had to simulate these cells as individual agents. So these are entities that are interacting with each other. We had the possibility of the cells having 4 different fate outcomes, and the cells could do certain things. They could proliferate, differentiate, They had physical spaces, and they were responsive to morphogens. And the morphogens which they were responsive to were an adaptation of the early morphogen structure I talked about before, where you have BMP and Wnt self-activating each other and it being inhibited by noggin and DKK. Himanshu programmed this into a agent-based model system. And here you can see what that would look like if I showed you the movie, where you start with a homogeneous cell and you end up with this wonderful pattern. And this pattern is exactly the same pattern you would get if you did a 1,000-micron colony, and different from a pattern you did a smaller colony, but completely predicted in silico. And of course, this then allowed us to do all kinds of wonderful experiments with the computer. If I can just figure out how to start my, I need to change this to a pointer. No, it's not working. Let me see if, oh, there we go. 1, 2, 3. So this is then what you're seeing are cells actually with that gene regulatory network in them, differentiating, responding to that environment, and patterning tissues based on the signals they receive from the environment, or the rewiring of that gene regulatory network structure. Now, I found that really exciting. And the reason I found that exciting is because what we're doing is we're taking the same cell and we're perturbing that cell based on knocking in or knocking out different components in the regulatory network. And we're getting completely different tissue patternings. Now, if you did that at the individual cell level, you'd always get the same thing. But the timing and the interactions between that tissue and its environment means that the predictability of the perturbations on the overlying tissue organization are more difficult to ascertain without having this interaction type model. And this is just a pinwheel to show you that. So we started with the pattern which I showed you at the beginning with no simulation. If you knock out these genes, you get these different patterns here. If you knock out these genes, you get these. And then if you overexpress or underexpress these inhibitors, you get these things. Of course, we've shown experimentally that our in silico model is actually highly predictive. These are a few examples here just to orient you. We have the in silico simulation with the control, the knockout at different times, and then we have the cell image, the micro image of the control versus the knockout in the different times, and then we have the quantification of those images. And what you can see just by looking at these two is that we're able to be highly predictive of how we perturb the regulatory network structure and how that affects the tissue overall organization. Of course, what's happening here is that the context of the gene regulatory network, which underpins the tissue development, changes as a function of position within the colony. If you're a cell that happens to be at the middle of the factor of the colony, if you get a certain gene knocked out, you have a different input, a different signaling input into that environment than if you're a cell out here. For example, here noggin and DKK4 are dominant, whereas here BMP is dominant over DKK4, and you get these different cell types, and all four combinations of cells occur here. And so because of that contextualization of the signaling inputs to the regulatory network structure is where you get that spatially differentiated tissue development there. Okay, so we're excited about this. We have a multi-scale digital twin to understand how cell regulatory networks can interact and start to predict this. And of course, we're starting to apply this and scale it to other tissue systems, especially blood development, which we're very interested in, and the emergence of blood cells out of the early hemogenic endothelial cell population. Just because I know David is interested in this, I'll extend some of this work to 3D tissue development and organoid systems, and that's really where we're starting to think about how to use to control symmetry breaking within aggregates or organoid-like scenarios. The experimental system, which is developed by Marina Bocek, is shown here. This is the AgriWell technology, which we developed in the lab some time ago by Mark Ungren. We actually place a BMP source, in this case, beads with BMP immobilized to the surface at the bottom of these AgriWells. That creates a seed point for this feed-forward autoregulatory loop, which creates gradients throughout these organoids. And then this allows us to then examine how different organizations might occur. So these are the different controls. If we have no local source, you get homogeneous differentiation. If you add just BMP to the media, you get this salt and pepper differentiation, as I showed you here. If you put beads without BMP on them, but if you put beads with BMP, you start to get this nice three-dimensional elaboration of the two-dimensional radial patterning that I showed you before. And of course, this is very much like the early convergent extension type patterning that you get during early development. We can do this at scale. So this is just zooming out and seeing hundreds of organoids of this type organized within this dish. And then if we look more closely at the underlying biology of this system, we can see that we have, we're starting to get nice organization of SOX2 brachyury and mesoderm with epithelialization of the early developmental cup there around where SOX2 is expressed. And I think I have this trilaminar internal structure really shown here, where you can see how this this pit where the cells migrate into as they develop into mesoderm actually occurs in these developmental gastroloids or organoids in these systems. So conclusions from this section, I think we have a nice platform to be able to predict and control spheroid formation. I really like the way that these developmental organoids are highly robust, and we're starting to think about ways to use these systems to recapitulate early developmental models, and this is really where I think it would be really fun to have a discussion. We're wondering what type of pathology actually might be evident so early on in development that we can start to study different things and use this as a model system for perhaps understanding some of those phases. Of course, as a lab, we're applying these systems to blood development. We're particularly interested in a phase of development that we haven't quite reached in our organoid system, but is probably developmentally a few weeks later, where you have the formation of the dorsal aorta, where on the ventral surface of the dorsal aorta, the hemogenic endothelial cells delaminate and bud and become blood progenitors or definitive blood stem cells eventually, which give rise to the blood forming system. Using many of the same technologies that I talked about earlier, we're able to recapitulate this process with a lot of control. Here are these early developmental organoids, in this case patterned to make hemogenic endothelial cells that are then seeded onto these nice 2D patches. And then when hemogenic cytokines are put into you can see them bud off the surface and form little blood progenitor cells. We can then take these cells and differentiate them using other types of systems into more mature blood cells. And I'm showing you here the generation of mature T cells. And in fact, we're taking this through initially this AgriWell induction, hemogenic endothelial niche, and eventually T cell development, largely activated by DL4, which eventually gives rise to these CD3 positive, TCR positive CD8 cells. So this system works very well. Where we're having trouble, or where we're focusing our efforts next, is although we're becoming quite good at generating mature CD8 positive cells from CD34 positive cells out of hemogenic endothelial cells derived from pluripotent stem cells, we don't yet understand well how the other lymphoid cells might be patterned from these cells. So this allows me to bring up one more project that might be of interest to this community, which is a wonderful collaboration between Fabio Rossi, Megan Levings, and my group, where we're starting to ask, how does this genetic information, how does the information on how T cells receive developmental signals pattern across tissues, in this case, the neonatal thymus, and can we use this information to help guide the types of signals which we would put on these cells in vitro to generate, for example, CD4 positive cells or other cell types that are currently more difficult to generate. So with Fabio and Megan, we're taking neonatal thymuses and we're doing a number of different analyses. We're doing site-seq analysis to link known cell surface receptor expression to the underlying gene regulatory networks. And then we're using Codex, which is a protein-based multiplex imaging technology to help us cluster and locate these phenotypes within a 3D tissue. And then we're doing, in collaboration also with Helen Blau's group, and we're doing some spatial interaction analysis of how cells are organized, that to try to understand what the key signals are within the niches that generate the types of T cells that we're interested in. So just some early data on this to finish up. We've used site-seq to start to identify both the different cell types as well as their organization. This gives us a map of cells as they proceed through these developmental loops. This is just an example of both the phenotyping and the functional markers which we're using within this system. This has now allowed us to take this thymic tissue, segment the different cell types, cluster them according to their neighbors, and recreate the thymic tissue using a fully computational elaboration. Now you see the medulla under this condition and the cortex and the different cell types within the cortex. And what's really exciting is then you can go in and you can say, well, what is this particular niche? This is a myeloid niche within the thymus that we were surprised to find, where dendritic cells, myeloid cells, and ICAM positive epithelial cells are interacting. Of course, we can then go in genomically and ask, what are these cells expressing? What is special about that niche, and how is that niche different to all these other neighborhoods which are here where these different cell types might be present? We can also go back to the images, and this is an image of an area around a vessel in which thymic progenitors are emerging. We've identified the vessel with smooth methyl actin and CD31. We can then go forward and look at what the signaling is with respect to collagen and JAG1 in that, and then further elaborate on that signaling to see how homing might occur with CCR7 or other chemotype signaling type receptors. We can then go back and isolate those and associate them with different cell types to try to extract what might be the underlying signaling interactions within this system. Where is this going? We're just starting to use this information now to identify how key decision points. So if you think about this loop here and this differentiation here, what you're doing is you're going from T cell progenitor. This is mostly the epithelial cell part of the thymus. This is the T cell developmental part of the thymus. We know that the T cell progenitors up here, we're able to recapitulate this, but there's also a subset of cells that we can't get to yet in vitro. We're starting to understand what the barriers are to that, and for example, different transcription factors or factors, and then we're adding these back to our cultures to see if we can use the thymus as a way to teach us how this development would occur. Okay, I'm gonna finish up there. I'm just checking on time. Really, so far I've talked to you a little bit about how essentially we're reverse engineering how tissues are developed. We're understanding the regulatory network inside individual cells. We're unpacking what happens in a tissue and starting to ask, do we understand it well enough that we can simulate it? Some work that I'm not talking about today is really about the forward engineering of that structure, where given this knowledge, can we actually impose certain patterns or certain outcomes on tissues using synthetic regulatory network circuits that are introduced to these cells, and actually program unusual tissue shapes, outcomes, or functions based on our understanding of that system. I've tried to acknowledge and really celebrate the great work of the students, postdocs, and research associates who did the work along the way. The lab itself is shown here on a retreat earlier this year that we were able to really get together for the first time in a long time and focus on things. And of course, I'm thankful for funding from a number of different organizations. David, I can stop and take questions here. I have a few slides on the school itself that are more kind of overview, but I don't know if you have time or interest.
It's really important, I think, for the department, particularly those who aren't really aware of what the School of Biomedical Engineering is. But if you go through those slides, then maybe we'll take questions on everything at the end.
Okay, sounds good.
Really appreciate that. Thanks.
Yeah. So now, as you can tell, I'm switching to the nice corporate slides, but really, I'm really happy to be able to, the School of Biomedical Engineering is about 5 years old. We started as a partnership between the faculties of medicine and engineering, and our goal really is to use quantitative engineering technologies to provide a technological foundation to biological innovation. And I think we've identified 3 major areas of focus for our early hiring. I'm not sure whether these slides are moving forward automatically. Cellular and molecular engineering, data and computational biology, and human interfacing devices. And we've been really lucky to recruit fantastic colleagues over the last 5 years in each of these areas, including folks like Ali Bashashadi, who's also a member of pathology. The school is really a comprehensive school. We have a graduate program that has several hundred graduate students in it. We have an undergraduate program focused on the same areas that I talked about for our research focus. And then we have an engineering design spine that goes through our curricula in both the graduate and the undergraduate program that leads to industry-sponsored capstone design projects and internships, and then at the graduate level, the engineers and scrubs program that some people might be aware of that we're continuing to develop. We're very excited also that we're the school, because it now has almost 400 undergraduate students as well as graduate students and growing faculty. We have a new building that we anticipate will be finished by late 2024, early 2025. And this is really to serve as a home for the school, but also a hub for interactions with our partners. In that school, we will have lecture theaters, maker spaces, biofabrication spaces, and ways to convert education, research, and innovation. We're also excited that we're continuing to work in the Biomedical Research Center, which is our current home, and that will become more of a core facility innovation hub, where new ventures emerging out of the school will be incubated, supported through new facilities. We're hoping that through the biomanufacturing initiatives that are underway, and perhaps even cell manufacturing facilities that are being planned through CFIs, that this will serve as a comprehensive home, really, for being able to take discoveries to first-in-human type trials over the next number of years. And maybe that gives a little bit of a background on the school itself.
Fantastic. Okay, so there are a bunch of very laudatory comments of people saying, thank you so much for an amazing talk, and people who've had to drop off early.